So welcome back again, whether you walked away and took a break to think about stuff or just to go to the bathroom, get a drink or whatever, or you followed on through the break to see what the answer to the concept check question is. Welcome back. So large numbers of NADPH and ATP are used in the Calvin cycle. We know it's 6 NADPHs and 9 ATPs. What does this indicate about the value of glucose as an energy source? Well, if you think back to cell respiration, and how many ATPs you get from a single glucose molecule, you hopefully recall that a single glucose molecule can yield 30 to 32 ATPs at the expense of 9 ATPs in the Calvin cycle. To make a G3P, and it requires 2 G3Ps to make a glucose molecule. So that's 18 ATPs to make a glucose molecule, from which you can extract 30 to 32 ATPs through cell respiration. So in essence, you get about twice as many ATPs per glucose molecule as it requires to make an ATP molecule. So let's fire, make sure the pen's working. So just putting the note in here. So again, if we think in terms of making a G3P, as we saw in the math problem in the previous section, it requires 2 G3Ps to make a glucose. It takes 9 ATPs to make a G3P. So we double down to make a glucose. That's 18 ATPs to make a glucose in photosynthesis. But in cell respiration, we can get 30 to 32 ATPs via cell respiration. So glucose as a source of energy provides twice as many ATPs as it requires to make it through photosynthesis. So obviously, glucose as a source of energy, exceptional source of energy in terms of energy budgeting. If you can double up the number of ATPs that you can get from a glucose molecule versus the number you need to make it, you're always ahead of the game. And remember, it's all about being ahead of the game. So long as the sum does not equal 0, that you'll always have a positive result in terms of the number of ATPs, you're always ahead of the game. And here, you're clearly ahead of the game. So what we need to do now is recognize that while C3 photosynthesis, what Calvin first identified, works exceptionally well for plants that are growing in temperate environments where it's not too hot, where the light is not too bright, where water is not an issue. C3 photosynthesis works exceptionally well. So in temperate environments, so say north of Florida, C3 is an outstanding way of carrying out photosynthesis. However, where it does get hot, where light is very bright, where water is an issue, C3 has a real drawback, and that drawback is a result of rubisco. So I've got to give you the name of rubisco one more time. It's ribulose bisphosphate carboxylase / oxygenase. And it's the oxygenase activity that is the issue here. So this process called photorespiration is really unfortunately misnamed. It's easy to understand why, because the process results in the loss of carbon dioxide, the exact opposite of what we expect to see in photosynthesis, where carbon dioxide ought to be taken up. And in fact, the loss of carbon dioxide or release of carbon dioxide looks a whole lot like respiration, hence the name photorespiration, because it does occur in the presence of light. But the problem is that Rubisco is attaching oxygen to the RUBP, not carbon dioxide. And as a consequence, you have a loss of energy, you have a loss of carbon dioxide, and it's an all-round negative effect. And this is particularly critical in situations where light levels are high, where it's hot, and where there's a restriction on water. And it all has to do with the preference of Rubisco for carbon dioxide versus oxygen. And this is why the carboxylase versus oxygenase becomes really critical. And you may ask yourself, why do plants even use an enzyme that can be affected by oxygen in such a central role in photosynthesis? And the short answer is because by the time oxygen, which remember is a product of photosynthesis, becomes a significant part of the atmosphere, the plants were already committed to using Rubisco and its carboxylase activity. Early on, being an oxygenase didn't matter. There wasn't any oxygen to interfere with photosynthesis. But later, as the plants themselves started, or cyanobacteria first, started to modify the atmosphere with the addition of oxygen, the oxygenase activity becomes quite the issue. And particularly for terrestrial plants, not so much for aquatic organisms, but it particularly becomes an issue for terrestrial plants because they're in the air, they're not surrounded by water, they don't have the cooling effect of being immersed in water, they don't have the sheer fact that they are immersed in water and therefore don't lose water. They have ready access to carbon dioxide because oxygen does not dissolve well in water, but carbon dioxide really does. So being immersed in water is not a big issue, but once you become terrestrial, oh, everything changes. Having a lot of light, having reduced carbon dioxide, having high temperatures, having reduced water, that all becomes a real issue once you become terrestrial. And yet we know that plants grow exceptionally well in those circumstances. When we think in terms of tropical rainforests, we think in terms of lots and lots of plant growth, but there we have high temperature and high light. We go to the desert. Deserts are not free of plants. Plants grow quite well in deserts. In fact, some plants will only grow in deserts. So how do plants get away with it? Well, obviously, there must be ways of modifying photosynthesis to take advantage of that. And there are two basic ways of modifying photosynthesis that we need to talk about. And both of them involve modification of leaves, as you'll see. So the first is referred to as C4 photosynthesis for the simple reason that here, the first product of incorporating carbon dioxide is a 4-carbon molecule, just as Calvin's C3 photosynthesis was called that because a 3-carbon molecule was the first carbon-containing molecule. Here, a 4-carbon molecule is the first carbon-containing molecule that we identify. It also involves not only a modification of the chemistry, but a modification of the structure of the leaf. So a classic C4 plant so typical, that's so ugly. Clean that up so you can read it. So C4 was first discovered in sugarcane. Now, you may not know this, but corn is actually not a naturally occurring plant. It's a product of man's breeding. It's actually derived from a little grass that still grows in Central America called teosinte. So corn, as we know it today, is actually a tropical grass. It's been artificially developed by selective breeding over many, many generations by people living in Central America. And it's a C4 grass. And the shape of a corn leaf, that long, thin leaf with the parallel veins running up it along the length of the leaf, that's a classic C4 plant, a grass leaf. Now, there are C4 plants that don't look like that, but for the classic ones that you're familiar with, the grasses are the ones that you'd be most familiar with. Sugar cane, put the E on here. So when we look inside the leaf, and to understand the difference, you might want to go back and take a look at the structure of the C3 leaf, because that's what we were looking at right at the beginning. So if you go back and look at the diagram of the leaf from the beginning with the palisade mesophyll and the spongy mesophyll, that's a classic C3 leaf structure. So here is what a corn leaf looks like in cross section. So you still have spongy mesophyll. You have running inside the veins, the vascular tissue, and the same is true with the veins in any leaf are where the vascular tissue is located, the water transporting and food transporting tissue. In a C4 plant, there is this layer of cells called the bundle sheath cells that surround the vascular tissue. And it's in here that the Calvin cycle occurs. So the Calvin cycle doesn't go away. It still occurs, but it has a specific location in the bundle sheath cells. So here you can see in the diagram the bundle sheath cell, and here's the Calvin cycle functioning. So why this is important is twofold. Number one, the bundle sheath cells are buried deep inside the leaf. So they're isolated. So they're isolated from O2 in the air, and they have no PS2, so no water splitting. And of course, if you cast your mind back to thinking about the light reactions, that's important because water splitting is associated with PS2, and water splitting is where we get the oxygen being produced. So the bundle sheath cells actually represent an O2 free zone. Which is perfect for the Calvin cycle and for rubisco. If there's no oxygen around, rubisco is only going to focus on carbon dioxide. But the problem is, being buried deep inside the leaf and isolated from the oxygen in the air, they're also isolated from carbon dioxide in the air. So there is a carbon dioxide pump that involves the mesophyll cells, the bundle sheath cells, and glycolysis. Yeah, it involves glycolysis as a way of doing this. So the carbon dioxide comes in, and instead of being bound to RUBP, it's bound to a molecule called PEP, phosphoenol pyruvate. Phosphoenol pyruvate is the second last molecule in glycolysis. It is converted to pyruvate by the removal of a phosphate group that in turn makes ATP. So we bind the carbon dioxide to PEP, which is always present in the cytoplasm because the cells are doing cell respiration. They're doing glycolysis. Plants do glycolysis, remember. And the enzyme PEP carboxylase only likes to bind carbon dioxide to PEP. It's not interested in oxygen. So it doesn't matter whether oxygen is present or not, nor does it matter if carbon dioxide is present in high levels or low levels. PEP carboxylase is happy binding carbon dioxide to PEP. That forms a molecule called oxaloacetate, which is in turn converted to malate. Now that happens to be the reversal of a step in the citric acid cycle. So there's an involvement of the mitochondria here as well. That malate then diffuses from the mesophyll cells into the bundle sheath cells through the plasmodesmata. You remember those, the connections between the cells in plants through the cell walls. The malate then has a carbon dioxide removed, a carboxyl group, because it's an acid. That carbon dioxide then enters the Calvin cycle, just as we've seen the Calvin cycle before. No difference. The remainder is then the pyruvate, which then diffuses back into the mesophyll cells, where the last enzyme in glycolysis converts the pyruvate back to PEP by burning an ATP, the exact reversal of the last step of glycolysis. So this is a modification of the fundamental biochemistry of the plant cells to allow this to happen. But what it means is that in a situation where there would be high light and therefore a lot of water splitting, meaning a lot of oxygen being produced, which would be the tropics, or where it is incredibly hot and there's the real risk of becoming dehydrated, which means you'd want to close the stomata to stop the water loss. But that means the oxygen builds up in the leaf and you reduce the amount of carbon dioxide because it can't get in anymore and the oxygen can't get out. The PEP carboxylase is quite happy to use just a small trickle of carbon dioxide to allow photosynthesis to continue because it's just not going to pay any attention to the oxygen. And of course, the bundle sheath cells are an oxygen-free zone. So this is the reason why tropical grasses like bamboo, like sugarcane, like corn, like wheat, like a lot of tropical grasses are C4 plants and grow exceptionally well in the bright light because they have the C4 pathway going on. And so they have the ability to process carbon dioxide without having to worry about being affected by the presence of oxygen. Because where the Calvin cycle is being carried out in the bundle sheath chloroplast, there is no oxygen. They're buried and isolated away from the oxygen in the air, and they lack photosystem 2, so they don't do water splitting. They're an oxygen-free zone. So that's the C4 pathway. When it comes to desert plants, They use a modification of the C4 pathway. It's called CAM, or crassulacean acid metabolism. CAM will be fine if that's what you want to learn. And here, they open their stomata at night because it's cooler. Makes sense to do that. And they capture the carbon dioxide at night. But their big issue, of course, is there's no light energy to create the ATP and NADPH necessary to process the carbon dioxide into sugars. So rather than opening their stomata during the day, they open their stomata at night, create the organic acids, the oxaloacetate and the malate, but store those. And they store them in the central vacuole. When daylight comes around and they have to close their stomata, well, no problem there, because they've got all this stored carbon dioxide in the form of the organic acid sitting in the central vacuoles. They mobilize that now and process it using the modified C4 pathway to release the carbon dioxide that enters the Calvin cycle, because there's now light energy to provide the ATP and the NADPH to do this. The pyruvate that's released as a consequence of all of this is then stored as glucose in the form of starch in the chloroplasts. That pyruvate is converted to glucose, and that glucose is then stored as starch in the chloroplasts. When night comes around, the starch is mobilized, the glucose is released, processed through glycolysis until you get to the PEP. The PEP is now available to bind carbon dioxide to, so that you get the oxaloacetate and the malate, which is stored in the central vacuole during the night until you have light energy during the day to process it. It is truly a modification of the C4 pathway. The big difference is this is taking place in two separate locations, and we refer to that as a spatial separation, two different locations. This is taking place at two separate times, or a temporal separation, a time separation. The basic idea remains the same. So here you can see a classic C4 plant, sugarcane, where we first discovered it. And this is a classic CAM plant, the pineapples. And you might wonder, why is a pineapple being identified as a potentially desert plant? And the reason why is because pineapples belong to a group of plants called bromeliads, and specifically epiphytic bromeliads that grow in trees. They don't use their roots for getting water and nutrients out of the soil. They use their roots for gripping onto the trees so they don't fall out. That's where the ancestors of pineapples come from. And so pineapples, as part of this group, do not use their roots for extracting water and minerals out of the soil. They get their water by capturing it in the cup that's formed by the leaves, where the pineapple actually, where you can see the pineapple sitting in the plant there. And they get their minerals in the rainwater and also in the dust from the air, at least the pineapple relatives do. Now, the pineapple as we know it is, again, a product of artificial breeding. You can find natural pineapples, but they're tiny little things like this. If you go to the Lou Gardens in Orlando, if you happen to be located in Orlando, if you go to the Lou Gardens, you can actually see the native relatives of pineapples, and you can see that they're tiny little fruit like this. We've artificially selected for larger, sweeter fruit that are edible over time. But again, pineapples do not like a lot of water around their roots. They do not like to get their feet wet. They actually grow in climates where there is very little water that falls as rainwater. So when we think in terms of the tropical locations where they grow, and think Hawaii, The area in Hawaii where they're actually growing is actually functionally a desert because it receives less than 10 inches of rain a year. It's on the wrong side of the volcanoes. So it doesn't get a lot of water, but it's perfect for growing pineapples as a consequence. You can grow pineapples here in Orlando if you want to. The actual next pineapple plant is sitting right here. It's the top in the pineapple that you buy at the supermarket. If you break that out and I'd plant it in a pot so that you can control how much water is there. Put it in a reasonable sized pot, keep it out in the sun, protect it from frost, and look after it for 2 years. That's right, 2 years, because it takes that long to produce a fruit. You'll get a pineapple. And if everything goes according to plan, you'll get a pineapple that'll taste like no pineapple you've ever had in your life. They are sweeter, juicier, and they just taste so much better than anything you can get from the supermarket, because the ones in the supermarket are picked green. They're not allowed to ripen on the plant. This is what a ripe pineapple looks like when it ripens on the bush. That you can never buy in the supermarket because it simply wouldn't tolerate being transported. When I worked for the University of California, we used to grow pineapples in our greenhouses so that we would have fresh, juicy, sweet pineapples throughout the year. Yeah, we used to do that. And let me tell you, it is absolutely worth it if you're willing to do it. But again, I digress. Ooh, I think I've done something wrong here, and am I doing the recording? I hope so. I'll find out when I'm done. Yeah, so this is the difference between the C4 and the CAM plants. They do things, one does it in different locations, one does it at a different time. So that brings us back to the diagram that we began with, showing the light reactions and the Calvin cycle with all the details filled in here now. So this is the diagram that is your starting point in understanding what goes on in the light reactions in the Calvin cycle. This should never, ever be your ending point. It is too simple, but it is a good starting point. Remember, you do need to know the flow of electrons in the light reactions, both cyclic and non-cyclic. You do need to know the basic Calvin cycle. Again, as a math problem is how I would look at it. Learn the math problem, not the full details as the book presents it. So the final slide is simply a summary of the light reactions in the Calvin cycle. Again, a fairly simple summary. Again, a nice starting point, not your ending point. You do need to know a lot more than this. So one more concept check. And again, we're not going to take a break this time, but even though I've got it here for a break. So one more concept check. It's been demonstrated that the bundle sheath chloroplasts only have photosystem 1 and totally lack photosystem 2. We talked about that. What effect would this have on oxygen concentration in these chloroplasts, and what benefit would this have for photosynthesis? So remember that water splitting is associated with photosystem 2. So if you don't have photosystem 2, you don't have water splitting. If you don't have water splitting, you don't make oxygen. So the lack of photosystem 2 means there is no oxygen in these being produced in these chloroplasts. What's the benefit for photosynthesis? The fact that the oxygen is no longer going to inhibit the role of rubisco in fixing carbon dioxide, because the oxygenase activity will never happen. There is no oxygen. So the rubisco is going to function as a carboxylase and constantly bind carbon dioxide and fix carbon dioxide in the Calvin cycle. So that's photosynthesis. Again, I've tried to outline for you all the things that you need to know through the concept questions and my discussions and outlining and filling in some of the details there and some notes that I've put on the slides. Again, as always, if you have any questions, concerns, or comments about anything that I've covered here, by all means, come and see me during office hours. At this point, as I said, we are finishing up the chemistry for this class. From here on, as we venture into the remainder of the chapters for the semester, we are not focusing on chemistry and chemical reactions and biochemistry. We're talking about cellular processes, not chemical processes in detail. But now that we've finished this chapter, we are now in a position to do the next exam. So the next exam comes up in a week. I know it's been only a little while since you did the last exam, but we do need to keep moving in order to make sure that we leave enough time to cover everything and fit in all the exams. So we do have the next exam coming up fairly quickly. But again, if you have any questions, concerns, or comments about anything that's been covered in this presentation, by all means, come and see me in office hours. Otherwise, I'll see you back here again when we start talking about cell division, which is the topic of Chapter 12. I'll see you again.